really interesting part of the world, and I'm only sorry I can't spend more time here. And thank you to the audience for staying. I'm not quite sure who I offended to be the last speaker, but it's great that there are people here. So I have worked on mutations and mutagenesis for a number of years, and I'm certainly conscious that one of the other speakers showed a family photo, and I'm going to do the same, because this is actually my own grandmother and her family. So this is my grandmother here. And the point about showing you my grandmother was not that she lived for 100 and so years, but that she actually only lived for 60 or so years because she died of gastric cancer at a relatively early age. Her mother and her sister developed colon cancer at around the same sort of age and also didn't have a very long lifespan. My grandfather and his second son developed prostate cancer, again at a relatively early age, and didn't have a particularly long lifespan. So as you'll realise, I have a vested interest in trying to work out if you can overcome cancer susceptibility which runs in families. So I certainly have a considerable interest in nutrient-gene interactions. Now, we know that environment plays an important role and that diet is a key part of that. But when I think back to my own grandparents' families and eating situations, I'm certainly not conscious of a really bad diet that was going on there. Perhaps they ate that when I wasn't present. But certainly gene-diet interactions may have been a part of it. What we know about cancer, it's a genetic and it's also an epigenetic disease, and there have been a large number of excellent presentations that have really highlighted this already throughout the conference. And what we know, and this is a schematic, and you've seen various versions of this Vogelgram, as it's sometimes called, is the schematic of this, that you've got mutations in key genes, and APC and KRAS, and tumour suppressor genes, which are involved in the progression. It's also clear, and there was an excellent session on epigenetics yesterday, that epigenetic events are vitally important in the progression. And so hypomethylation and hypermethylation are likely to be key events, as are also other sorts of events that regulate epigenome. And so DNA damage and other events lead to aberrant gene expression and promotion of enhanced proliferation, and these mutations may decrease the function of tumour suppressor genes or increase the function of oncogenes. And so we've known these sorts of things for an awfully long time now. What's clear then is that much the same as heart disease, that cancer can arise directly from genetic perturbations. They can arise directly from genetic perturbations interacting with metabolites from various dietary components or environmental components and increase the risk of heart disease or cancer. But what the increased evidence suggests is that we've got gene pathways that are acting along with environmental or dietary components, along with genetic susceptibility, to lead to heart disease and cancer, a sort of hierarchy of causal relationships that seem to be increasingly important for common human diseases. Now, one of the things that I think becomes a little bit murky is the fact as to whether it's the genes that are acting directly to increase susceptibility and leading almost directly to cancers. And so some of these sort of genetic components like BRCA1 and BRCA2 may be directly leading into breast cancer. Others of them may be leading to mismatch mutations in colon cancer, but it's also clear that cancer susceptibility is associated with families, and identical twins have genetic susceptibility, and so if your twin has cancer, you've got a 20 or 30% increased chance of getting cancer. So both direct genes going through to cancers directly, but also susceptibility genes, and it's these susceptibility genes that are almost certainly interacting with dietary components to increase cancer risk. Okay, so these sorts of genes are affecting cell cycle control. They're affecting signals from these sorts of receptors that others have talked about in this conference, regulating, again, cell cycle control, angiogenesis, tumour invasion, and TP53 causes apoptosis, cell cycle arrest. So well-recognised mechanisms and clear understandings increasingly have been developed over the last 20 or so years as to how these genes can get involved and regulate cell cycle control and cancer progression. Now, I'm going to spend the rest of my talk thinking specifically about one type of cancer, because if you look at the cancer incidence figures, New Zealand is often amongst the highest countries in the world, and certainly at the moment, the most common cancers in the world are lung, breast, bowel, stomach, and prostate, and we're up there amongst the top figures. Breast cancer high in New Zealand, but we're not the highest in breast cancer, but at the moment the incidence of prostate cancer 25 times higher in Australia and New Zealand than in South Central Asia. And so here's the current, the most recently released incidence figures for prostate cancer, and here we are right up there leading the world. So as you'll realise, this isn't a figure we're particularly proud of, it's a figure we'd like to be able to think about how we can approach doing something about it. So where are the carcinogens? If we're thinking about cancer, we're thinking about things that cause cancer, and where are the carcinogens and how do we find them? Well, certainly when you look at the nature of cancer and the involvement of mutations in those progressive steps, it's reason to believe that they would be mutagenic, and mutagen testing has been an early approach to picking these up. And so I'm certainly conscious that some of my own early work was influenced quite strongly by the sort of work that was set up by Bruce Ames in California, where they were looking at mutagenicity testing as a means of picking up carcinogens. And so this is Bruce Ames in his 80s now, still going strong. And this is the test system that he developed, a bacterial test system, which of course isn't a human test system, isn't even an animal test system, but nevertheless led to a number of chemicals, hundreds and hundreds, thousands and thousands of chemicals being identified as suspect carcinogens. Now, what was clear when you took some of those things that were identified as mutagens and you put them into carcinogenicity tests, that it became clear that an awful lot of components of the diet actually caused cancer in rodent carcinogenicity tests. And so here is just a range of examples. Fungal toxins weren't included, but you name it, virtually anything, any dietary component that you can find comes up as a rodent carcinogen. Now, this sort of information was starting to come out, and I think this table was produced in the 1990s, and it leads to a sort of almost silly situation. What do? Do you essentially go to a situation of deciding that eating may not be good for you and avoid virtually everything in the diet? Well, there's some truth in that and the whole obesity crisis. And so here, I gather this is a real place in the States. The heart attack grill, taste worth dying for. And there's one of their satisfied customers. And as you'll realise, this sort of meal is not just a heart attack, but a cancer risk that's waiting to happen. So obesity and excess food consumption, there's no question that's associated with a number of chronic diseases. And so the obvious situation here is to actually decrease the amount of food consumption. But it's also clear that there are specific types of dietary items that may be important both as cancer causes and cancer prevention. And stepping back and working out which of those lists of hundreds of dietary chemicals are likely to be important is certainly been an initiative in a number of labs. Now, as I mentioned, it's quite clear that it's not diet alone, it's gene-diet interactions that are likely to be important, and certainly I think much of the work in the last 20 years has focused on understanding the nature of events in cancer and thinking about what sorts of genes might be important, so candidate gene-diet interactions. And so people have focused on genes for xenobiotic metabolism, DNA repair. We know if we get a DNA adduct, a DNA lesion, that there's a probability that that is either repaired or that cell cycle control genes, that cell is killed by apoptosis rather than carrying through to a mutation in the DNA. We know these genes interact with mutagens, and we also know that they interact with anti-mutagens. There's quite a close balance of things that cause cancer, but also protect against cancer in the diet. And I think John Milner talked about a number of the anti-mutagens that the NCI is particularly interested in. So, which types of cancer-causing agent and dietary component are likely to be really important? Are we likely to be consuming at levels that really are likely to impact? So moving from animal studies into human studies. Now, flame grilled meats is something that came out in the 1970s as something that caused a mutation. And as you can imagine, this affected the heart of every American. The American hamburger was seen as a potential cancer hazard. This information reached New Zealand, and certainly some of my own colleagues in the 1990s got together and asked the question as to whether high temperature cooked meats and fish were likely to be a problem in the New Zealand diet. And so here's the heterocyclic amines formed during high temperature cooking. They react with DNA to form DNA adducts and lead to a number of changes. And so they can affect tumour suppressor genes, they can affect oncogenes. And so here is our own publication, and I should say that was one of our early publications in the area, and it was a whole exercise in logistics. We did some cooking of meat in the New Zealand population. We attempted to export them to the United States. And so carefully cooked, precisely cooked to define temperatures were actually seen as a threat to the American citizens, and so FDA got these arriving in the States and promptly destroyed them. So not to be outdone, we felt that we had to do something to make sure we got there, and we discovered that there was a wrinkle in the regulations that meant that if we personally escorted cooked meats to the United States, we were going to be able to get them into the country in order to test them. So my next trip to the United States, I went accompanied by 10 kilograms of cooked meats in my hand luggage. It created a degree of interest, and I won't even attempt to report the discussions with the customs officers, but we did manage to get them into the States, and we were able to show that we got heterocyclic amines produced in the New Zealand meats. They are somewhat different concentrations, different things from the American situation, but approximately doubling the prostate cancer risk of New Zealand men who eat regularly high-cooked meats in the diet. So reason to believe this is a real cancer hazard in the New Zealand population. Now, what we know about heterocyclic amines is that they're metabolized in various ways, and depending on the genetic component, they're either metabolized into a form in which they will react with DNA, or they're metabolized into a form in which they will be excreted in the diet. And so that the relative balance of these processes depends very much upon the background genetics of the individual. And so everybody didn't have a doubled risk of prostate cancer. Some people would have had a much higher risk, and some people wouldn't have had any risk whatsoever. They can carry on eating their barbecued meats at the New Zealand sort of summer barbecues. And just to give an example of the last 2 years alone, There are literally hundreds of reports of well-done meat consumption and various metabolic enzymes in various population groups. No question, but a really well-characterized gene-diet interaction that really does imply what you can think about in terms of modifying cancer risk. Now, the good news, of course, is that you can actually protect against dietary mutagens, and I was one of the authors in what's a very useful book that was produced last year. And so just to give you an example of these, and I'm not going to go laboriously through all of these, we've already heard about glutathione transferases, detoxification enzymes. So it's a good idea to have these enzymes functioning correctly, and you want them more efficient than the enzymes that will create DNA reactive compounds. But a number of us in the population do have a gene deletion in that particular enzyme, and that's associated with increased prostate cancer risk, but this is where it starts becoming complicated. So well-done meats, if you've got that deletion, you're going to have an increased prostate cancer risk, but you can overcome that by high consumption of cruciferous vegetables. These are rich in glucosinolates, which are precursors of sulforaphane, and via a series of events that they can actually overcome. In the absence of these, they can actually overcome and lead to excretion of the particular offending mutagens. So well-done meat, if you've got the right genetic background, it's not going to be a problem. If you haven't got the right genetic background, lots of cruciferous vegetables. And just increasing the rate of excretion, but a high consumption of cruciferous vegetables may compensate. So diet interactions associated with a genetic composition. And as John Milner said, just this one seems to be really quite an important genetic mutation. We've looked and masses of studies about dietary mutagens, but I think it's important to recognise that the same sort of mutational consequences can also occur as a result of dietary deficiencies. And so a lot of work, particularly Jim McGregor in the States, Michael Finneck in Australia, Bruce Ames, have particularly shown how folate deficiency and other sorts of deficiencies can lead to chromosome aberrations absolutely comparable to things like ionizing radiation. And so just to give examples, lots and lots of different types of deficiencies, and a number of them associated with increased prostate cancer risk. Vitamin D deficiency, folate deficiency are things we're particularly concerned about in the New Zealand population. We also don't get enough selenium and probably don't eat enough omega-3 polyunsaturated fatty acids. So quite a number of these are probably relevant to the high prostate cancer risk in our country. And also in the US population, it's not just the New Zealand population that's deficient in these, and certainly folate fortification has been thought to resolve the low folate problem, but as John Milner showed you, there's now concerns that there may be some increased risk, and probably just for a part of the population. And almost certainly, where you're starting to see some of those risks occur, it may be people who've got variants in one carbon metabolism, and others have gone through this pathway. And it seems that low folate is a particular problem for those who've got a common polymorphism in the MTHFR enzyme. If you've got adequate dietary intake, it acts as a valve, and you've got this change in the pathway. But if folate's poor, then that variant promotes the misincorporation of uracil into DNA, and you get chromosome breakage, you get mutations. And so if there is actually a problem with parts of the population, it may suggest that this is one of the key genes where we should be getting genotyped, and personalised nutrition may be preferable to folate fortification in this particular case. Now, genome-wide association studies have come up at a number of times in this conference, and I'm certainly conscious that they've changed our thinking. a lot of the work in prostate and other cancers, we've worked on what we understood, and we've certainly been proved to be right by basic understandings of cell cycle genes, folate metabolism genes, and other sorts of things. And when we've gone one by one, nutrient by nutrient and gene by gene, and looked for interactions, a lot of our predictions have come true. But of course, as with many other diseases, genome-wide association studies are not hypothesis-driven and have led us to understand the nature and pick up genes that I certainly didn't even know existed. And so let's just have a look at what you might, 2 years after the first of the major genome-wide association studies with prostate cancer have come out, and have a look at some of the major genetic polymorphisms that we now see as being implicated in the risk for prostate cancer. And so we'd suggested gene diet interactions, and we'd come through. And so here's the GSTM1 gene that we were talking about, and we can probably overcome that with cruciferous vegetables intake. But the p-value for this, it's not spectacular. It's significant, but it's not spectacularly significant, and it's a bit different in different populations. And so here's the MTHFR, the other one that I sort of identified, and again, we can overcome that with folate consumption. But again, our probability of it being a real risk and a high risk in the populations we're working with, it's not spectacular. But look at some of these other genes that, as I said, I didn't even know existed. HNF1 beta. Here's a probability value of 1 by 10 to the minus 15. JASF1, again, I didn't know the gene existed. And here, one study, there's not a fantastic association, but here's another study, about 8 by 10 to the minus 11. And again, these are genes where the evidence seems to suggest, when you start to look at the biochemistry and what's coming out about them, and what was known but not seeing them as primary genes, is that there is evidence of nutrient regulation of these genes. So I just wanted to look at a couple of examples and just try to work those through in the remaining time. So just here's this HNF1 beta, and it's a transcription factor, and we've heard about transcription factors and the way in which these are often nutrient regulated. It regulates insulin expression and other proteins involved in glucose metabolism. And it also has a downstream effect with this serine protease, and what's probably happening here is that you've got an indirect mechanism, and it leads to the sort of things that we were originally seeing as suspicious: cell cycle arrest, angiogenesis, cell migration, but it isn't a direct pathway, it isn't directly leading to it, and so it wasn't a gene that we were immediately looking at as being causal. Here's another one, going backwards there. Here's the other one, JASF1, a transcriptional repressor of testicular receptor. And again, it regulates serum glucose and IGF-1 levels, and really sort of links back with a number of the other sorts of things that have come through as themes in this conference. Again, associates with glucose and insulin secretion. And again, an interaction, an indirect effect that may be affecting mitogenic activity, cell cycle control, cell proliferation, and again by some of these other mechanisms. So again, that's just two examples, and there's a lot of them coming out where these genome-wide association studies are implicating genes that when we start looking at them, we recognise there are clear gene-nutrient interactions that we weren't even thinking about at an earlier stage. So I think these have been important and certainly are revolutionising the way we're thinking about cancers. So just thinking about prostate cancer and other cancer risks, if we could, it would be great to be able to choose our ancestors wisely. But we don't usually, or we haven't learnt at this stage to do that. So I think there's a clear message coming out that we need to understand our genomes, we need to understand the key pathways, and I think it's pathways that are going to be important, and I think it's increasingly clear that we need to learn which are the key genes, which are the key pathways, and how we should personalise nutrition according to genotype. Thank you.